Modern chemistry doesn't advance just by making new substances, it advances by knowing exactly what those substances are. At scales too small for human senses, chemists still need certainty: what elements are present, how much of each exists, and how atoms are arranged. Analytical chemistry is the reason this invisible world becomes measurable, reliable, and controllable. This is the Learning Studio, where chemistry is explained as the tool that turns uncertainty into knowledge. And after understanding how materials are engineered at the atomic level, the next step is to understand how scientists actually see and verify matter that cannot be observed directly. Most people think analytical chemistry is just lab testing, but in reality, it is the science of translating invisible molecular information into readable signals. For example, when a blood test detects disease markers, when food is tested for contamination, or when forensic labs identify unknown substances, analytical chemistry is working behind the scenes, converting chemical identity into data humans can interpret. At the molecular level, the mechanism is elegant. Analytical techniques probe how matter interacts with light, electricity, magnetic fields, or other forms of energy. Every molecule has a unique signature: how it absorbs light, how it vibrates, how its atoms respond to magnetic fields, or how its fragments move in an electric field. By measuring these responses, chemists can identify substances with extraordinary precision, even in trace amounts. This ability developed through the work of scientists like Robert Bunsen, who showed that elements emit unique spectral lines when heated, allowing chemical identification through light. Later, Felix Bloch helped establish nuclear magnetic resonance, a technique that reveals molecular structure by probing how atomic nuclei behave in magnetic fields. These ideas turned analysis into a window into molecular architecture. As analytical chemistry evolved, techniques like spectroscopy, chromatography, and mass spectrometry allowed scientists to separate complex mixtures, measure concentrations accurately, and determine molecular structures in detail. This transformed chemistry from a science of inference into a science of confirmation. Claims could be tested, purity could be verified, and reactions could be monitored in real time. The impact of this capability is everywhere. Pharmaceuticals rely on analytical chemistry to ensure safety and correct dosage. Environmental monitoring depends on it to detect pollutants at parts per billion levels. Materials science uses it to verify composition and defects. Even space exploration relies on analytical instruments to identify compounds on other planets. Analytical chemistry also enables progress across all other branches of chemistry. Without accurate measurement and identification, reactions cannot be optimized, materials cannot be trusted, and discoveries cannot be verified. It acts as chemistry's quality control system, ensuring that what we think we've made is actually what exists. And once we understand how chemists see and measure the invisible, the next question naturally follows. If molecules can be identified and measured so precisely, then how can chemistry be applied to understand and manipulate the chemistry of life itself? That question takes us directly into the next episode, where we explore biochemistry and the molecular chemistry of living systems.